Welcome to Avatar Technology Digest. Get ready for exciting news on technology, medical cybernetics, and artificial intelligence. And here are the top stories of the last week. A controversial European neuroscience Blue Brain project aims to simulate the human brain in a supercomputer. It has published its first major result, a digital imitation of circuitry in a cent-grain-sized chunk of red brain. The work models some 31,000 virtual brain cells connected by roughly 37 million synapses. The goal of the Blue Brain project is to build a biologically detailed computer simulation of the brain based on experimental data about neurons, 3D shapes, their electrical properties, and the ion channels and other proteins that different cell types typically produce. Researchers who created this simulation said its digital neurons flicker with activity seen in real rat brains and replicate patterns of electrical behavior that are triggered, for example, when a rodent's whiskers are touched. The simulation draws on 20 years of measurements from rat brains and took a decade to turn into code that runs on an IBM Blue Gene supercomputer capable of solving billions of equations every 25 milliseconds. The model can be manipulated in ways that are difficult to do experimentally, providing insights into how individual cells contribute to the functions of neuronal networks. Many say that the Blue Brain project is overly ambitious, effort to reverse engineer the rodent brain and ultimately the human brain with a view to building a working model of the organ in a computer. Do that, the argument goes, and scientists might have a hope of understanding the neural mechanisms that underpin such extraordinary phenomena as consciousness, love, and the antics of the Bullingdon Club. Machines with artificial intelligence have shown quite an improvement in several aspects. For instance, they can play chess, compute complex mathematical problems, and even identify pictures. Researchers from the University of Illinois did a study on the ConceptNet 4 version of the AI, and they discovered that its IQ level is similar to a 4-year-old child. The Massachusetts Institute of Technology has created an intelligent machine called ConceptNet under its artificial intelligence program. The research team chose an IQ test known as the primary scale of intelligence. The IQ test is commonly used in schools across the United States and is designed to measure the level of intelligence in 5 key categories. The first one is information, in which participants need to answer simple questions such as where can you find a giraffe? The second category is vocabulary, the third is word reasoning, the fourth category is similarities. The final category is comprehension, where participants need to answer questions in the form of an explanation. Researchers found that ConceptNet 4 handled vocabulary and similarities well. In the information category, ConceptNet 4 got an average score, while it got poor scores in comprehension and reasoning. The ConceptNet scored a mark that is average for a 4-year-old child but below average for 5 to 7-year-olds, concluded the researchers. The result was highly dependent on how the intelligent machine understood and interpreted the questions. Age-related macular degeneration could be treated by transplanting photoreceptors produced by the directed differentiation of stem cells, thanks to findings published today by Canadian researchers. ARMD is a common eye problem caused by the loss of cones. The team of researchers of the University of Montreal has developed a highly effective in vitro technique for producing light-sensitive retina cells from human embryonic stem cells. This method has the capacity to differentiate 80% of the stem cells into pure cones. Within 45 days, the cones that we allow to grow towards confluence spontaneously formed organized retinal tissue that was 150 microns thick. This has never been achieved before. In order to verify the technique, researchers injected clusters of retinal cells into the eyes of healthy mice. The transplanted photoreceptors migrated naturally within the retina of the host. Researchers have been trying to achieve this kind of trial for years. Thanks to the simple and effective approach, any laboratory in the world will now be able to create masses of photoreceptors. Even if there is a long way to go before launching clinical trials, this means in theory that these findings will lead to effective solutions for countless patients. For years, scientists have been trying to manipulate pig organs so they could be transplanted into the 8,000 humans waiting for a life-saving transplant. So far, that hasn't worked, but George Church, a geneticist at Harvard Medical School and co-founder of the biotech company, hopes to change that. Earlier this week, he announced that he and his colleagues had used the gene editing tool CRISPR to modify an unprecedented number of genes in pig embryos in order to make them easier to transplant into humans. The one other big concern is rejection of donor organs by the human immune system. Church has reportedly tackled that problem too by modifying over 20 genes in additional embryos that make the proteins that irritate our immune cells. Researchers in China have also had recent success in making multiple CRISPR edits to pig genomes. They were even able to combine the technique with somatic cell nuclear transfer without mosaic mutation or any of the usual undesirable off-target effects. Perhaps the most exciting news from the Chinese pig geneticists has been their creation of custom pet rainbow micropigs. Not only are these pigs miniature due to inactivation of one copy of the growth hormone receptor gene, but they can be ordered in different colors. Church and his startup company eGenesis hope to begin implanting gene-edited pig embryos into mother pigs as soon as possible. DNA is your blueprint, firmware, and operating system all rolled into one, so of course it's really important for the code to be correct. But the nature of chemistry is that things sometimes go wrong at random. DNA breaks down over time, sometimes there are mistakes in transcription and ultraviolet radiation, and some chemicals can damage DNA. The Nobel Prize in Chemistry 2015 awards 3 pioneering scientists who have mapped how several of these repair systems function at a detailed molecular level. Now, thanks to Thomas Lindahl, Aziz Sankar, and Paul Moldridge, we have improved understanding of how our own cells work and repair damaged DNA. That, in turn, can help develop more effective medical treatments. Scientists worked independently over the years rather than collaborating on a shared project. Thomas Lindahl demonstrated that DNA decays at a rate that ought to have made the development of life on Earth impossible. This insight led him to discover a molecular machinery, base excision repair, which constantly counteracts the collapse of our DNA. Aziz Sankar has mapped nucleotide excision repair, the mechanism that cells use to repair UV damage to DNA. The cell also utilizes nucleotide excision repair to correct defects caused by mutagenic substances, among other things. Paul Moldridge has demonstrated how the cell corrects errors that occur when DNA is replicated during cell division. This mechanism, mismatch repair, reduces the error frequency during DNA replication by about 1,000-fold. That is all for Avatar Technology Digest. Thank you for watching us, and of course, you're welcome to subscribe, follow us on social media, and join the global conversation. See you soon.